Hello students, in this video we will discuss about the photophosphorylation. What is photophosphorylation? The process of formation of high energy chemicals like ATP and NADPH. The process of formation of high energy chemicals ATP and NADP in presence of light. In the presence of light, it is called photophosphorylation. These are non-cyclic photophosphorylation. Photophosphorylation. Non-cyclic. Here is given in your book Z schemes, also called Z schemes of light reactions. This is light. From the light the photons comes and the LHC, light harvesting complexes. The electrons evolve and the electrons accepted by the electron acceptor, and after that electron comes to electron transport systems. These electrons comes in the LHC again, and also here is a change, ADP plus 1 phosphate, that is converted into ATP. Adenosine diphosphate converted into adenosine triphosphate. Here electron accepted by LHC, that is electron acceptor. It is in photosystem 1, the photosystem 2, and this is the photosystem 1. In 1 also NADP converted into NADPH. Okay. This is Z schemes, and two phosphosystems work in series. First PS2 and PS1. These two photosystems are connected through an electron transport chain, that is this one, also called Z schemes. This is Z. And both ATP and NADP plus H are synthesized by this process, and PS1 and PS2 are found in lamellas of grana. Hence this process is carried here. Okay, this is all about the Z schemes. Now the cyclic photophosphorylation. Cyclic photophosphorylation. What is this? Cyclic photophosphorylation, only PS1 work and electrons circulate within the photosystem. This is given in your book. Cyclic. This is cyclic photophosphorylation, light and chlorophyll P700. Here the electron transport systems, after the accepted by electrons by electron acceptor, and again comes into the chlorophyll. This is a cycle complete. Photosystem 1 only is present. The electrons circulate within the photosystem. It happens in the stroma lamellae. Possible location, because in this region PS2 and NADP reductase enzymes are absent. Okay, and has only ATP molecules are synthesized. It occurs when only light of wavelength beyond 680 nm are available for excitation. Okay. Now what about the electron transport system, ETS? The electron transport in Z scheme. The electron transport in Z scheme. In pH2, reaction center chlorophyll absorbs 680 nm wavelength of red light, which makes the electron to become excited, and these electrons are taken up by the electron acceptor, that is passes them to an electron transport system, ETS, consists of cytochromes. The movement of electrons is downhill. Then the electron passes to PS1 and move downhill further. Now the splitting of water. Okay. It is linked to pH2. Water is split into H plus and O, and electrons. It means 2H2O gives 4H plus O2 plus 4 electrons. Okay. Now what about the chemoosmotic hypothesis? Chemiosmotic hypothesis. What is this? Chemiosmotic hypothesis explained in the mechanism of, this is diagram given in your book. Chemiosmotic hypothesis explains the mechanism of ATP synthesis in chloroplast. In photosynthesis, ATP synthesis is linked to develop of a proton gradient across the membrane. Here is the proton present and proton gradient is developed. Okay. From where these protons comes by the splitting of H2O. And ATP synthesis linked to development of proton gradient across the membrane. The proton are accumulated inside the membrane of thylakoid in lumen. And ATPase enzymes has a channel of that allow diffusion of proton back across the membrane. This release energy to activate ATPase enzyme that catalyze to formation of ATP. This is the formation of ATP by the ADP and P. Okay. Now the biosynthesis phase in C3 plant. Biosynthesis phase in C3 plants. Here is a diagram given in your book. This is also called Calvin cycle. Here there are three main steps: carboxylation, this is carboxylation, it is reduction, and also regeneration. Now what is the carboxylation? CO2 combined with the ribulase 1,5 biphosphate, this is, and the atmosphere CO2 plus H2O combined with, it is called carboxylation. By the carboxylation 3-phosphoglycerate is formed, and after that the second one step, that is reduction. Carbohydrate is formed at the expense of ATP and NADP. Here is ATP plus NADP gives rise ADP and P plus NADP. It means it is used. This is called reduction. And the third one is regeneration. By the reduction, triphosphate is formed, that is sucrose and starch. After that is converted into sucrose, that is after that starch. Third step is regeneration. The CO2 acceptor ribulose 1,5 biphosphate is formed again, and ATP converted into ADP. This is the third step. Okay, you can write properly. Number 1, carboxylation. Number 2, reduction. And number 3, regeneration. Okay. Now ATP and ADP are the products of light reaction are used in synthesis of food. The first CO2 fixation product in C3 plants is 3-phosphoglyceric acid or PGA. The CO2 acceptor molecule is RUBP. Full form ribulose biphosphate. The cyclic path of sugar formation is called Calvin cycle, on the name of Melvin Calvin. On the name of Melvin Calvin, it is called Calvin cycle. The discover this pathway, who discovered? Melvin Calvin. And 3 main steps of the cycle. Now what is the 6 CO2 plus 6 RUBP plus 18 ATP plus 12 NADPH. What gives out? 6 C6H12O6, that is glucose, plus 6 RUBP plus 18 ADP plus 18 P inorganic phosphate plus 12 NADP. These are the products. What we can conclude. What are input? So we can say input 6 CO2, 18 ATP, 18 NADPH. And what's output? 1 glucose, 18 ADP and 12 NADP. Okay.